Hi, my name is Kaylee Bernardo, and I'm going to be talking about neurons, action potentials, and how they actually work and communicate with other cells in the body. A neuron is an electrically excited cell that communicates with other cells surrounding it. It makes up the nervous system, and this is actually one whole neuron right here, and there's a bunch of different parts to it, so I'll go through those right now. The dendrites are these little finger-like projections on the ends of the cell body. Dendrites receive messages from other cells, so they will receive the stimulus and collect everything, and they're very sensitive to the stimulus that it is around. The cell body, or also known as the soma, is kind of like the basket that's collecting all of the stimulus. This cell body collects all of this information from each and every dendrite and holds it within here. The axon is actually the bridge, basically, that connects the that sends along the message to the cell from the cell body to other neurons or muscles or glands. In this picture right here, in this case, it's to another neuron. So once all the cell body collects all of this stimulus from the dendrites, it can trigger an action potential once it hits a specific threshold, which we'll talk about in a little bit. But an action potential is an electrical signal that travels down an axon. It's really important that there's myelin sheaths along these axons as well. Myelin sheaths insulate the axon so you don't lose that action potential, because right here this is a long way to travel for an action potential, and you want it to stay constant. You want it to stay activated and triggered. So myelin sheaths are really important to help out with that. And actually the degradation of myelin sheaths is actually the cause of the disease MS. That's the whole reasoning for it. It's because it's hard for the action potentials to be held because there's no myelin sheaths on here. There's also the terminal buttons, which are at the very end of the axon. These will actually form junctions with other cells. So like I said over here, the other cells like neurons, muscles, or glands. And then right here is just another dendrite or another neuron that we can see is going to be receiving the signals from this neuron once it sends an action potential to the ends. So there's a few different types of neurons in the body. There's sensory neurons, interneurons, and motor neurons. The sensory and motor neurons are part of the peripheral nervous system, and the interneurons are part of the central nervous system. So in this diagram right here, we can see that this sensory neuron is inside the skin, and it will receive stimulus. It'll feel that, the hair follicles are moving, or it'll feel that, someone touches you or something, and it will send an action potential. Once it's triggered, it will send an action potential down into the interneuron. So this is it's the collection of the stimulus, the receptor. Once it gets into the interneuron is like the integration center. It's where the information that the sensory neuron gave it is integrated and then processed into how to react to this sensation that we got, into this stimulation. So once it figures out, what to do with this, information, it then sends another signal into the motor neuron, which the motor neuron will then go and communicate to the effector, which could be a muscle, a gland, something like that, in order to respond to this stimulus that we received by the sensory neuron. So the actual chemical way that action potentials are produced is a little complicated, but also I think is very interesting. The resting potential of a neuron is negative 70 millivolts, and this is a constant negative 70. It's always at this, around this, during the resting potential, and the way that it regulates it is there's an ion pump that pumps sodium and potassium ions in and out of the cell to keep the cell at this negative 70 millivolts. It's not shown in this picture, but it is there, and that's what the whole purpose of it is to maintain that resting potential. In this picture right here, these are actually voltage gated ion channels. So these ion channels are activated by a change of voltage, whether it's increasing or decreasing, and I'll get into that right now. So in the resting state, all of these voltage gated channels are closed because there's no change in the voltage, anything like that. It's just at negative 70 within the cell. So the cell is more negative than the outside of the cell at this point. Once there is a little bit of stimulus, which is right here in number 2, the ion channels start to open, or more and more of the ion channels start to open, specifically the sodium ion channels. So as we can see right here, only one of them is open, and that's because there's just a little bit of stimulus that the neuron is receiving, and because the inside of the cell is negative, once it opens, the sodium ions are going to want to rush into the negative neuron. Once enough of these rush into this negative neuron, that's when you hit the threshold right here. This negative 50 millivolts is the threshold that triggers an action potential, an all or nothing action potential. So once this neuron is negative 50 millivolts within inside of it, then we're going to see this next step right here. So during this next step, once we hit that threshold, all of the sodium channels open. It's not just a few of them or, none of them. It's all of them open at once. And since this, the inside of the neuron is still negative, because we're at negative 50 right now, all of them are rushing inside of the cell. So this is a very fast action. The sodium ion channels open and close very fast. So they will, and you can see here that the potassium channel remains closed right now. So as the sodium ions are rushing inside, this is depolarizing the neuron, and it's causing this huge spike up right here, this number 3, to where you get to this tip right here, which is, around a little bit less than positive 50 millivolts. Later on, the potassium channels are actually slower in opening their gates than the sodium channels are. The sodium channels are really fast and open and close very fast. But the potassium channels are kind of a little slower at it, and they open them and then they wait a little bit and then they'll close them eventually. So this downfall right here, this phase number 4, is this step right here of the potassium channels opening and the sodium channels closing. So now we know that the inside of the cell is positive, and now the sodium channels are closed. And once the potassium channel is open, the potassium ions are going to want to rush outside of the cell now, because it's going to be attracted to the negative outside of the cell. So you can see that's why there's this downfall right here, which is called repolarization. And so this action right here is going to cause the cell to get more and more negative again and head back towards its resting potential. Eventually, the potassium channel closes, but like I said before, it's very slow at opening and closing. So it's going to close very slow, and this dip right here is the reasoning why. Is the reasoning why. Because it closes so slow, the uh, the neuron undershoots and gets actually a little bit more negative than its resting potential of -70. And then eventually the potassium channel closes, and then it gets back to its resting potential state again through its flow of potassium and sodium channel, that it's not the ion uh, gated channels. It's just going to be this constant flow of pumping the ions in and out to maintain that -70 millivolts again. Now, when an action potential, so this is all happening, it's getting sent down the axon right here, sent down the axon into the terminal buttons. And this little section right here is what we're looking at right here. So the action potential will arrive into the terminal buttons, and it will actually, it's not in this picture right now, but it causes, this action potential causes calcium gated channels that are on this terminal button to open, and calcium will go into the cell. This calcium going into the terminal buttons causes the vesicles to fuse with the plasma membrane and release the neurotransmitters into the synaptic cleft. The synaptic cleft is where all the neurotransmitters can kind of travel from one cell to the other. Right here, this is another receiving neuron that's going to be getting all of these neurotransmitters bound onto its ion channels. So these neurotransmitters are released into the synaptic cleft and bound on, the neurotransmitters are then bound onto these ion channels, which cause the ion channels to open and causes ions to rush in or out of the cell, which can depolarize or repolarize the cell, causing it to fire more often or fire it less, which it more often is an excitatory neurotransmitter, or else a less, cause it to fire less, which is a inhibitory transmitter. Also, which isn't shown in this picture, is the reuptake of these neurotransmitters. These neurotransmitters, once they're kind of done being bound onto these ion channels, they will get reuptake back into this terminal button, which is very important because there can be a lot of issues right here if there is no reuptake to it, or if there's too much reuptake of it. So yeah, I just think the neurons are very interesting, and thank you for listening.